The central problem of evolution concerns the origin of information and meaning in biological systems, especially in light of the Cambrian explosion and the role of horizontal gene transfer. What is at stake is not merely how organisms change over time, but how biological processes come to function as information-bearing systems rather than as undirected chemical reactions. The question is not simply how life evolves, but how life comes to mean anything at all. Traditional evolutionary theory has tended to locate the origin of biological information in self-replicating molecules and to explain subsequent complexity through random mutation filtered by natural selection over vast timescales. By the Cambrian period, however, this explanation reaches its limits. RNA and DNA are already fully developed, highly structured systems capable of encoding, transmitting, and executing instructions. The deeper puzzle is no longer replication, but interpretation. How do molecular sequences reliably result in specific functional outcomes rather than chemical noise? Modern molecular biology makes this problem unavoidable. DNA is no longer understood merely as a chemical substrate, but as a genuine coding system. The nucleotide bases, adenine, cytosine, thymine, and guanine, function as an alphabet. They do not directly produce proteins. Instead, they encode instructions for producing them. In this sense, DNA is not merely analogous to code, it is code, instantiated in chemistry. This reframes a core assumption of classical Darwinism. Random mutation plus selection describes one mode of change, but it does not capture the dominant mechanisms now known to drive biological innovation. Horizontal gene transfer fundamentally alters the evolutionary picture. Through conjugation, transformation, and transduction, organisms, especially bacteria, can acquire and execute genetic instructions originating outside their direct lineage. In conjugation, a donor cell extends a pilus to a neighboring cell and transfers a single-stranded segment of plasmid DNA. In transformation, cells absorb free DNA from their environment. In transduction, viruses act as genetic couriers. Together, these mechanisms allow organisms to download and deploy fully functional genetic modules rapidly, sometimes in minutes or hours. Adaptations that would be astronomically unlikely to arise through stepwise mutation can spread across entire populations almost instantaneously. This process is not driven by altruism or foresight. In pilus-mediated transfer, the receiving cell typically plays a passive role. The regulatory trigger often resides within the plasmid itself. Many plasmids are self-propagating genetic elements, equipped with switches that activate transfer machinery under stress conditions such as antibiotics, crowding, nutrient limitation, or DNA damage. The donor cell retains its genetic material by replicating what it sends, while the recipient synthesizes the complementary strand and gains a fully functional plasmid. The apparent precision of the exchange arises not from intent but from selection acting after transfer. Plasmids carrying useful traits persist because their hosts survive and reproduce. Plasmids carrying useless or harmful genes disappear. A clear illustration is antibiotic resistance. Resistant bacteria can rapidly endow neighboring cells with resistance genes, transforming entire microbial communities in short order. What might appear as gradual adaptation becomes population-wide change on compressed timescales. Evolution in this context is no longer a slow, solitary climb, but a network process of information exchange. The significance of this mechanism expands dramatically when framed through the work of Carl Woese, the Nobel Prize-winning biologist who identified archaea as a distinct domain of life. Central to Woese's broader vision was the proposal that during the earliest phases of life, when organisms were exclusively single-celled, horizontal gene transfer may have been the dominant mode of evolution. Rather than evolving along isolated ancestral lines, early life formed a communal genetic network in which innovations were shared, recombined, and redistributed. In this view, modern genomes are not the product of a single lineage, but a deeply integrated mosaic assembled from countless organisms across deep time. This network model resolves a central difficulty in classical evolutionary thinking. Vertical mutation alone would require immense stretches of time to generate complex novelty. Horizontal gene transfer compresses those timelines by allowing organisms to acquire fully formed functional modules rather than building them incrementally. The difference is akin to one programmer attempting to generate a complex program through random keystrokes versus a team transferring a complete working software package. Entire blocks of genetic code can be rewritten and deployed at speeds once thought impossible. Crucially, this exchange is not chaotic. DNA behaves less like noise and more like language. It operates under strict syntactic constraints that govern which nucleotide sequences can yield functional proteins. Just as grammar constrains meaningful sentences while permitting enormous expressive freedom, molecular biology operates within structural rules that allow for creative recombination without loss of coherence. New biological functions emerge not by chance alone, but by recombining meaningful units within an established grammar. Genes are therefore rarely transferred as isolated fragments. They move as expression cassettes, packages that include not only coding regions but also promoters that initiate transcription and terminators that bring it to a close. This ensures that transferred genetic material is executable upon arrival. The system is built not merely to transmit sequences, but to deliver usable instructions. Yet this only sharpens the deeper question: how does genetic information function as information in the first place? The ribosome does not produce random proteins. It produces specific proteins that correspond exactly to the RNA message it receives. This implies interpretation. RNA acts as a messenger, but the ribosome must read that message in a rule-governed way. Without interpretation, there is no information, only signal. Meaning, therefore, appears to be fundamentally relational. Information requires a transmitter and a receiver. DNA functions as a speaker. The ribosome functions as a hearer. RNA mediates between them. Meaning does not reside solely in molecules, but in the structured relationship between encoding and interpretation. This reframes the search for the origin of information. Rather than locating meaning exclusively in DNA, attention may need to shift toward the interpretive machinery that renders DNA intelligible at all. This relational understanding also reshapes evolutionary metaphors. The traditional tree of life, branching lineages diverging through vertical inheritance, fails to capture the reality revealed by horizontal gene transfer. What emerges instead is a web, intersecting pathways, loops, feedback systems, and shared genetic pools. Evolution proceeds not as a linear ascent, but as a communicative network. Where there is communication, there is meaning. Seen in this light, the Cambrian explosion ceases to be an anomaly. It becomes the natural outcome of a biological system capable of high-speed information transfer, modular recombination, and rapid deployment of functional instructions. Evolution accelerates not because chance increases, but because information flows. This reframing leads to a final open question. If meaning in biology is fundamentally relational and networked, then prevailing paradigms of meaning itself may be incomplete. Meaning may not arise from simple correspondence between symbol and object, but from patterns within a web, more like a neural network than a dictionary. If life is structured as a communicative system, then understanding its origins may require rethinking not only biology, but the very nature of information and meaning itself. Credits: research on pili and plasmid, an interdependent transcription model by David Carter. Acknowledgement to Perry Marshall from Evolution 2.0.